Great.
Awesome. So hi again, everyone. Great you're still with us. And I'm very happy to introduce Eva Dyer to you. Eva completed all of her degrees in electrical and computer engineering, including a PhD, master's, and bachelor's. The former two at Rice University and the later at University of Miami. And she's currently an assistant professor at the Wallace H. Coulter Department of Biomedical Engineering at the Georgia Institute of Technology and Emory University, where she runs the Neural Data Science Lab, where she and her team develop new machine learning and data science approaches for making sense of large-scale neural networks. We're all super excited for your talk, and please take it from here, Eva.
All right, great. Thank you so much for the invitation. We were originally going to be in Montreal at this point, so I'm a little sad that we can't all be together, but I'm very excited that I have on unicorn slippers. So I haven't ever given a talk in such comfort. So so I'll go ahead and get started. My talk today is about work that we've been doing in basically taking neural recording. So I'll be talking about some kind of similar topics to what Carson talked about last time. So I'm really pleased that she did a great job of kind of setting the stage for a lot of the topics that I'll be discussing. So yeah, we're talking about taking neural recordings and being able to compare them or think about how they might vary or change across different types of perturbations to the nervous system. So whether it be across time, across space, so different subsets of neurons is what I mean in this case, or across different behaviors as well. So a lot of the work in my lab is motivated from the sort of emergence of many new techniques for recording high dimensional and very high resolution neural activity and also brain structure. A lot of this has kind of come to be over the past decade or so with the Brain Initiative, and these major advances have produced really high resolution and high dimensional data, much like what Carson was talking about in the previous talk, with these larger data sets, right, where we either have recordings from more neurons or where we have higher resolution of images that we've used to capture brain structure. In all these cases, we need to think of ways to actually make sense of all this data. And so my lab works on a number of different strategies for taking high dimensional data and making sense of it. And often that does involve finding lower dimensional formats to put the data into. So a question we are often faced with is why reduce dimensionality? And so there are a number of different rationales, different reasons depending on the application. In one case, obviously if we take something that consists of a lot of information, right, a lot of bits, a lot of neurons, or a lot of time points, and we can boil it down into a smaller set of numbers, then this allows us to effectively compress the data. And this is, the kind of core backbone of MP3 or any sort of compression algorithm, is actually finding a new basis or a new representation of the data that allows it to be re-represented in a lower dimensional or compressed format. In addition to compression, which is a kind of obvious one, there's also advantages of reducing dimensionality in terms of denoising. So essentially the idea is that if the data has sort of low dimensional structure to it, then when noise is corrupting it, this will often kind of move it off of this low dimensional subspace. And so by enforcing this low dimensional structure, we can actually get rid of a lot of noise or artifacts potentially. And then the third reason why we'd like to reduce dimensionality is often for interpretation of complex data. And I think that Carson did a really great job of setting the stage for that. Ultimately, we're faced with a question of whether or not we can understand what these complex systems are doing. And oftentimes when we refactorize or represent our data in a lower dimensional space, in some cases, we can actually place meaning or interpretation to those lower dimensional representations. So just to sort of set the stage for the rest of the talk so that we're on the same page, I'll be sort of thinking about general tools for dimensionality reduction, or many of the things I'll describe, could be applied to different types of data, which, generally speaking, would be represented, let's say, as a matrix, where we have a number of different features, and then we have different samples, in this case, along the columns. And so in the examples that I'll show right after this, and the sort of core motivating example, we're also thinking about a similar set of features as Carson's talk. So now each of the different rows essentially is a different neuron. And now we're tracing out the neural activity of each of these neurons over time. And that's essentially one example of this matrix that I'm describing here on the left. And now what we can think about is essentially representing now each column of this matrix, which is a point in time, and that represents the activity of, let's just say, D different neurons within our system. We can essentially represent that now as a point in this high dimensional space. Here I'm showing it in, a sort of three-dimensional view, but this is actually a higher dimensional space, but it's hard to, of course, visualize. So now each of these time points is now a point in this high dimensional space. And the key insight in low dimensional models and dimensionality reduction in general is that natural signals tend to not occupy every possible position within this high dimensional space, but oftentimes these data are sort of clustered or might live on these subsets within the larger D dimensions. And so one way to represent that is, I've already said the word, but is this idea of a subspace, which is what I'm showing here on the bottom, which is essentially fitting a linear or a planar surface to this scatter of all these different points in the high dimensions, right? So when we do things like principal components analysis or other types of linear matrix factorization techniques, we're essentially finding this linear approximation to this collection of data. Of course, the same idea can be extended to when data sets have nonlinear structure in them. So instead of having, this piece of paper or a plane, we can actually sort of fold this nonlinear structure around the cloud of points as well. And in this case, the term for this are manifolds. So I've written here manifold models. That sort of refers to these nonlinear subspaces of the space, of the larger space. All right, and there's many different extensions of these sorts of ideas depending on the sorts of low dimensional structures that one might expect to find within their data. I'm sure many of us are very familiar with the idea of clustering data, and we can also think about, if we look over here on the bottom right, looking at this cluster model, we can also think about it as itself a low dimensional model where we're essentially representing this collection of points now with their centroids. And so this gives us a form of sort of compression and re-representation. And on the left, I'm sort of just showing a hybrid of all of this, where we would consider a case where we actually, instead of having one subspace or one manifold, we might have a mixture of these subspaces or manifolds. And I'm describing this model because we'll see that it becomes useful in our later discussion, this union of subspaces model. All right. If there's any questions at this point, I don't know, Pierre, just, I guess, chime in and let me know if there are any questions in the chat.
So far, we have none. I will let you know once something comes up.
All right, cool. So yeah, so we sort of set the stage for this. The sort of general idea is, we take recordings, in this case of neural data, we find some low dimensional representation of it with a linear or a nonlinear technique. And then when we have this low dimensional or representation in this latent space, so to speak, we're often now faced with the sort of next question, which is, how can we take these two different distributions of activity or distributions in this latent space, and how might we be able to compare them across, say, two different data sets. So this arises in many different neuroscientific applications, and of course in many other areas of machine learning. Some examples are, I have a collection of data, before a disease onset and then after, for instance. That would be a comparison. We could also consider the case where, the first distribution is from a single time point and the other distribution is now from a subsequent point in time. So in all of these cases, we're often faced with this situation where we've gone through the process of finding a low dimensional representation of our data, and now we're interested in how these two data sets are actually related to one another. And in many cases, while PCA is one example of a matrix factorization technique that is unique, the actual representation of these data in this low dimensional space is not guaranteed to be the same. So what I mean by that is that, in many cases, we might find something similar to what I'm showing here, where we have a rotation or where we have some affine transformation of an original data set, and then what we see in the next data set is, a transformation of the original. And so in this case, we'd like to ask the question of how can we actually figure out a transformation that would align these two different distributions or two data sets, and that will then allow us to tell more concretely about how they've changed or how they've been altered across these different states. So really the goal here then is to figure out this function that will, bring these two data sets or distributions into alignment. So we can actually write this out or formalize this sort of idea with the following problem statement that I have here, which we'll refer to as a distribution alignment problem. So essentially what this is saying, so on the bottom I show here sort of visual for the high level strategy, which is we don't know the color of all these points, right? We don't know that the red correspond to the red and the blue correspond to the blue. We just have the shape of these distributions in this case. So what we'd like to do is be able to find a way to essentially, figure out that the blue and the blue should be connected and the red and the red should be connected to one another, or our corresponding points within these two data sets. And so the formulation that we have here on top is essentially we have mu, which is our source distribution. And I'm sorry I don't have a pointer. I wish I could kind of interact with this a bit more, but so we have mu, which would be our source distribution. We have nu, which is our target distribution. We're going to apply a transformation t to mu in order to align it with the target nu under some distance, which I've written here as math or subscript d, right? Or this divergence metric d. And we're also going to constrain the possible class of transformations to lie in a set t in this case. So this is a very general problem statement at this point. Hopefully it's clear. Our idea is to learn a t that brings mu and nu into alignment with one another. So there are kind of two main design variables in this problem here that we need to think about. The first is, which class of transformations we should consider. Obviously, considering all possible transformations makes this very difficult. In addition to the class of transformations, we're also faced with a question of which distance we should use to actually form or solve this alignment problem. And so to answer the second question about the distance, we've chosen a distance that comes from the literature on optimal transport. It's called the Wasserstein distance. I'll explain to you a little more in the application why the Wasserstein or why optimal transport distances are favorable, not just for this problem, but for sort of general transfer learning problems more broadly. But just to provide a little bit of intuition about what it actually is. So, many of you, I'm sure, are familiar just with the normal notion of a norm, or sort of the Euclidean distance between 2 points. And so if we consider here now the distance between these 3 different distributions here on the bottom, we have a blue, a purple, and a red. So because these 3 distributions are perfectly non-overlapping, then if we measured their distance in a normal sort of Euclidean norm or any other norm, we would say that they're all actually orthogonal to one another, right? Because they don't share any overlapping support. And that's sort of shown here with this triangle on the left, denoted LP, just that all of these distributions are equally far from one another. However, if we sort of think that the position in space is actually important for telling us information about nearness or closeness between these distributions, then we actually would want to potentially say that the blue and the purple are closer to one another than, say, the blue and the red, which are much further away from each other in terms of this axis, or in terms of the support that we're showing here. And so this sort of notion of geometry of closeness can be captured by the Wasserstein distance. And that's what I'm showing over here on the right, which is that the blue and the purple now are much closer to each other in terms of their Wasserstein distance than, say, the blue and the red. All right, so we can kind of break this down a little bit further. So this is just the definition of the Wasserstein distance, and in particular an entropically regularized version, which makes the complexity much better, and basically convergence is much quicker as a result. So here what I'm showing is just the definition of the optimal transport distance. So what you see is that it feels very complicated, because now all of a sudden, to even measure the distance between these 2 distributions, I actually have to solve an optimization program. But as you'll see, when you use this entropically regularized version, the updates are actually quite simple, and so this can be computed efficiently. So I'll just kind of step through what it is, because I think there's some nice intuition here. So essentially, your Wasserstein distance between 2 distributions, P and Q, is given by basically the minimizer of this program. So what we're doing is we're looking for a matrix P, which tells us how we're going to transport mass from distribution P to distribution Q. So P is basically the main operator that tells us how these 2 distributions are related to each other. And we're going to essentially multiply the entries of this transport matrix by the cost to transport between those 2 different bins. So we essentially give a cost associated with moving mass from bin I to bin J, and we're going to penalize now that transport based upon that distance. And in our case, we can just use the Euclidean distance in space as our sort of cost. So that means that when we have to move much further away in space, we're going to pay a higher cost. And then what we see here on the right is this entropy regularized term. And so this will essentially help to smooth out our problem, and we can think about it as a regularization term that also can in some ways be thought of as a way to help avoid overfitting in some cases. Okay, we need to make sure this is a valid transport plan, and so these are the constraints essentially to make sure that this is a doubly stochastic matrix. And then here, this is the entropy term, just writing it out. And now this just provides a little bit of visualization, or hopefully some intuition about what this entropic regularization does. So essentially what we're showing here on the left is if I'm trying to transport mass from P to Q, right, and they're shown along the rows and the columns, the optimal transport, or the transport plan with no entropic regularization, is shown on the left. And then as we increase the amount of entropic regularization, as we move to the right, what we see is that basically the transport plan is being smoothed. And that smoothing is actually very beneficial in terms of solving this efficiently. All right, so just recall, going back to our original alignment problem, which we see here on top, we had these two questions. One was which types of transformations should we consider, the other distance. So far, I hope I've convinced you, at least at this stage, that optimal transport can make sense when you have a geometric sort of, some geometry underlying the sort of space, right, over which you would want to compute distances. And then the second question is, which class of transformation should we consider? And so this in general is a hard problem, and it's very data set dependent often, and much of the field of transfer learning, which tackles these sorts of problems, is very specific depending on the types of shift across different data sets. And so we will then turn to our neuroscience application to really help us constrain the space over which we're going to delete. I guess this would be a good point to stop if there was any questions about the math or the sort of methods up until now. Okay. Great. So then, yes, so let's turn to our application. So in this case, what we're looking at is a situation where we can, implant an electrode array and measure the activity of individual neurons within different parts of the cortex. In this case, we're looking at primary motor cortex, which is, involved obviously in coding movements. And so we're recording neural activity while we're also monitoring the motion of the individual's reaching movements. So basically there's a cue for the individual to make a reaching arm movement to one of 8 different reach directions, and then there's a center point. So this is a kind of standard center out reaching task, as it's called. So what we're doing is we're able to both see neural activity simultaneously while we can actually monitor the kinematics or the velocity of those different reaches into different directions in space. So we have a 2-dimensional behavioral readout along with the neural activity. And a sort of general question that we're interested in with these sorts of data sets is what's called neural decoding. So basically finding a mapping between neural activity and the movement. And traditionally, this is done by, recording both of these things simultaneously, finding some mapping between the two, say on training data, and then using that, on a test data set after you've learned that relationship on the previous data. So in this case, what we can actually do is we can, and in some of our previous work, we've shown that you can take the activity of these neurons in motor cortex, right? You can reduce them down to a lower dimensional space, which is what I'm showing here on the bottom right. So this is after applying a linear dimensionality reduction technique, fact analysis, to embed the data into 3 dimensions in this case. And so we know that without any labels, without any correspondence between the neural activity and the movements, we know that the shape of the movements are actually preserved in the neural activity. So we were interested in using this sort of alignment method that I've described to think about how we can bring this high dimensional, at some point, neural activity down to a low dimensional space and then align it with this movement distribution, which I'm showing here on the left. And if we can do that effectively, we're able to essentially solve this neural decoding problem that I've described, but to do it without needing correspondence between the movement and the neural activity. This would then allow someone to potentially decode from a subject that they might not have captured training data from directly, but they know that the subject is actually doing the same underlying task that should have the same representational structure. All right. So based upon this sort of observation and the motivation of our problem, where in this case we have structure within the neural data that has clusters, right? Or this sort of union of subspaces model that I referred to earlier. We have that activity of neural populations when the subject is moving towards a particular reach direction tend to be clustered in this latent or low dimensional space. And based upon that, we basically have this clustered representation that we can exploit within our formulation. So I'm running time. I spent a lot of time going into details on OT. So I might kind of move quickly through some of this, but essentially the idea just at a high level, if we take our original optimal transport alignment problem, and now we're going to split it into essentially trying to find across clusters as well as find correspondence across individual points. And so by breaking this in this way and solving it in these two different stages, it both effectively aligns with less data and also avoids a lot of the local minima within this problem. And that's why I call it hierarchical, because it's sort of this two-tier approach. And so what I'm showing here is just the new formulation, similar to what we've shown before, but now we've essentially split our Wasserstein divergence, but now over these transformations of the individual clusters. So I'm just showing here, the breaking down of the source and the target into clusters. And now our objective is now going to be aggregated over all of these different alignments. And finally, in the end, we can enforce constraint, a constraint that all of the TIJs, so all of the individual transformations, have to agree on a global transformation. And that's just one assumption of our model. But one might also potentially extend this to consider a case where you could have different transformations for different clusters, potentially, if that's how you expect your data to be shifted. Here, we're also constraining T to be the class of unitary transformations. So in this case, we can find basically a rotation of our data that helps to bring these things into alignment. So the key contributions of this work, which was a paper at this past NeurIPS, was this hierarchical formulation of OT. We also have a distributed solver. We have both MATLAB and Python implementations if you're interested in testing it out. And we also show improved performance in neural distribution alignment. This is just some results on synthetic data showing you that HiWA, which is our method, hierarchical Wasserstein alignment, which is the blue on top, provides major benefits over just a simple Wasserstein alignment. So meaning if you weren't to leverage this clustered structure within the data. What's nice is that this, in a synthetic 2D case, we can show that using optimal transport over just a KL divergence, which was the original divergence measure that we used here, really smooths the landscape, and it reduces a lot of the local minima that the algorithm can get trapped in. So this is a really important part of the solution, both using optimal transport as a law, and also leveraging clustering structure when it does exist within your data. These are some examples and results of now trying this on two different non-human primates. So on the left, I show, one, and on the right is another. Here, because both of these individuals are carrying out the same motor task, which is constraining their neural activity to be low dimensional and have similar structure, we can actually now take neural activity from one animal that we haven't actually formed any target data on, or we don't have the movement. We can align it onto a target data set from a different individual. And so in this case, given that the constraint on behavior is consistent, we can actually find a cross-subject alignment in this case. This is showing that this is robust to subsampling. So within the macaque motor cortex, we find that, even though we have 150 neurons, we don't actually need that many to be able to decode reliably. And in some cases, when good subsets of neurons are selected, then you can do a lot better. And so maybe we can use Carson's method to find the good sets of neurons. We have also now extended this to to rodent cortex. So in this case, this is a striatum, a collaboration with Keith Hengen's lab at WashU in St. Louis. Here we're showing now 2 different data sets that are acquired over 2 different 12-hour blocks. And in this case, we see over periods of the animal being awake or being in 2 different stages of sleep, we actually find very consistent and reliable sort of geometry or shape across these low dimensional embeddings. And this allows us to then align these 2 and provide, a good way to then bring these data sets back so that any labels that you've collected in terms of sleep on a previous data set can then be transferred onto this new one. And we compare here with canonical correlations analysis as well. So I know we're running out of time, so we can play similar games here. Robustness to subsampling is lesser. We also have fewer neurons. So we're sort of interested in looking at how all of these methods apply when you're, obviously not able to acquire the number of neurons that, might be involved in this full circuit. And so I think that this is, I have, just 5 minutes left, right? So I think this is a good place to stop. Actually, okay, I'm just going to give you a little teaser. I wanted to talk about this. And I just wanted to quickly tell you about this other story, but it's related in the sense that, there are many cases where you might not already have a good feature space to work with. So in our previous case, we were able to take individual neurons over time and use that as the basis for solving this problem. But in many cases, especially when you're dealing with images, you might not already have a good feature space. And so deep learning has emerged as a powerful technique for feature extraction and representation learning within high dimensional image data. And I just wanted to tell you quickly about some work that we're doing where we call it deep brain disco for deep brain discovery. Essentially, the idea is to start by training a neural network to discriminate different brain areas using small local snapshots of the underlying anatomy. We train the neural network to solve this discrimination task, so pretty standard convolutional neural network. And then what we go in is we actually go into hidden layers within the network and use now the network as a feature extractor to tell us more about the data than what it's actually been trained to do. So we use the network as a feature extractor and then use dimensionality reduction on top of that to provide interpretable views into the underlying microarchitecture and anatomy within this data set. And we can use that to start to discover some interesting features of the data. So on the right, I'm just showing examples of manually annotated layers within our data and how our network is able to actually pull out these different lemma differences and start to group different regions within our sample that it hasn't been trained to do because it's essentially learned a rich set of features that describe the underlying structure in the data. And we can also pull out some fun things like, finding high cellular density regions also without training the network to do so. All right, so in summary, I told you about work that we've been doing using dimensionality reduction and also this optimal transport alignment technique to be able to do things like movement decoding in non-human primates and also decoding sleep and wake within rodent. And then at the end, I told you about some work for in using deep learning to learn representations to then find patterns and discovery in neuroimaging data. And we also have a new X-ray micro CT data set along with that for the community, along with annotations to help with other people developing machine learning methods on sorts of data. All right, and of course, the team that made it happen. So the main core work has been done by a student in my lab, Beja, as well as John Lee, also at Georgia Tech. And then the later work was Aish Balwani in my lab. And then we have amazing collaborators. I talked about some data from Keith's lab and our funding sources. And with that, I will end the presentation and take any questions. Thank you.
Thank you very much, Eva, for this very great and informative talk. I guess there's nothing like embeddings, dimensions, and alignments on a sunny Friday. That was a very great explanations as well. Really enjoyed it though. I searched for a disco emoji, but there unfortunately is none. So please just think about it. I was about to reply for your disco, deep brain disco thing.
I had one on my slide.
Oh,
I had one. I'll share it with you. Yeah.
So we have two questions I think we can just briefly address. It wouldn't be that bad if we go 2 minutes over time. We have one question from Roberto asking, is this method suitable for generalization across subjects? Does it need a high number of data points?
Yeah, that's a great question. So, I did talk a little bit about this result, right, where we could actually show a cross-subject alignment, taking a movement data set from one non-human primate and now taking a neural activity from a completely different subject, right, and finding a way to align the two. But of course, that relies heavily on the sort of task structure being sufficient to sort of constrain these neural activity patterns to be similar, right? So, in the case of the types of transformations that we use, right, we are assuming certain sorts of transformations, namely that they're not skewing or stretching the data. So, if I imagine that in a lot of other application settings across subjects, if you don't have enough to constrain them to be similar enough, you're going to see sort of a shift across domains. And in that case, incorporating those additional constraints could be added into this sort of OT framework. But currently, we don't incorporate kind of more sophisticated types of transfer.
Great, thanks a lot. And we have a second question.
Yeah, I see here. OT works with statistical distributions. How do you go from brain data to distributions? So, so what I've shown here is essentially taking, neural activity by time. And what we first do is we find a low dimensional representation of that data. Using something like PCA, or if it's nonlinear, using isomap, for instance. And so the method that I'm describing is working directly in the latent space. And now in the latent space, you're essentially, let's just say it's a 3-dimensional space. You now have the probability that you're going to have mass at a particular position in the 3-dimensional space. And the key idea is that, the first dimension for data set 1 might be a different set of dimensions, right? And so even though we have good representations of these two data sets in the low dimensions, we might need to now find correspondence across their PCs. Okay. Hopefully that answers the question. I think maybe the internet connection was not great.
Yeah, I mean, that's the beauty of it. Like, we can stream it on YouTube, or it will be streamed in parallel on YouTube. Then we're going to upload the videos after cutting them to talk specific sections. So if anyone, like me, for example, experienced connection problems, you can check it out later on the YouTube channel. So with that, I think we would call it your talk right now. Thank you very much again, Eva, for this amazing presentation. And
yeah, thank you for the invite.
It was our pleasure, really, though. And now we're going to head over to the next session. That is, I'm closing this down. I'm going to invite you over to see all of you there in a bit.